Hello, everyone. Thank you for the opportunity to present our work at this Museomics workshop. My name is Petra, and I'm a postdoc at the Wellcome Genome campus here in Cambridge in the UK. And what I would like to talk to you about today is how we adapted a double-stranded Illumina library prep approach for sequencing of old pinned fly specimens. So the collections, the types of samples that I'll be focusing on are historic, archival insect collections. Now, insects are the most abundant by biomass land animals. And for us humans, we typically tend to put them in little categories depending on how much they affect our lives. So we like to categorize them as disease vectors, as pests, as pollinators, as decomposers. So some of these are positive and some are viewed pretty negative. And so these are really important historic collections because we can see how these species changed morphologically. And by the end of this talk, I will also hopefully convince you genetically. And what is even more interesting is how many of these fall into the group of flies. Many fly species can be found across all kinds of niches. And humans tend to not like flies very much, and very often will use control measures such as insecticides to get rid of the ones in red, but unfortunately that also means that it will affect the ones in green. So what are the flies that I will be mostly focusing on and that we work primarily at the campus in our group? These are Anopheles mosquitoes, who are the key vectors in human malaria transmission. Anopheles mosquitoes, there's many species. They can be found all across the globe. And a subset of that are vectors for human malaria. So if you look at the phylogenetic tree here, all of the ones represented with a small square are major malaria vectors. And so in the past 50 years, one of the key players in stopping the spread of malaria and reducing mortality was the use of insecticides. The problem with that is they've been in use for so many years and so many new insecticides have been introduced. And so there's been a widespread insecticide resistance problem, which is a giant hurdle in future malaria control. And here we get to our project, which we dubbed Neanderskeeto. So if we look at the present day population genomic databases that we have, we can see that depending on the region in the world, depending on the species, we have different insecticide resistances, which are very often population specific or region specific. But when did this exactly start? So if we go back 100 years ago, there were no specific vector control measures. You could drain the swamp, you could use general poisons, which would affect not just insects, it would affect also other animals like livestock and humans. And so in the 1930s, specifically 1939, DDT, which was discovered already at the end of the previous century, was found to be a very efficient insecticide. And so in the 40s, it was widespread use in Europe and in the US, in agriculture primarily, but it also helped eradicate malaria in many of those regions. And then in 1955, the World Health Organization started its malaria eradication program, and that started the deployment of insecticide-treated bed nets and indoor spraying of insecticide all across the globe, primarily sub-Saharan Africa. So we have this time frame within the last century where humans have exerted a really strong evolutionary pressure on these mosquitoes. And so the question is, how exactly did this affect the populations? And lucky for us, people have been collecting malaria-transmitting mosquitoes for the last 100 years, since it was discovered that mosquitoes transmit malaria parasites. And these can be found in historic archival collections. The adult mosquitoes are typically pinned and dried. And here on the left are just 2 boxes from the London Natural History Museum, which houses 2.5 million fly specimens. Just a very small subset of that are actually mosquitoes. And on the right here, we see a little lady from this London collection, which was collected in Kenya back in the late 20s. And so everything you get from these specimens is usually found on these tiny labels that are attached to it. You're hoping that those labels will at least have location and date, preferably also be stored in a box with other samples of the same species, and then you can select samples that match what you're looking for. Preferably, but that's rarely the case, some entomologists have also done proper field studies and field books of these samples, so you can get a bit more data on other things. But at the end of the day, you're mostly working with what's on this tiny label when pinned insects are in play. And so, as you can see, these are extremely tiny, extremely valuable samples. And so curators are very reluctant to give such samples to biologists, to geneticists who would normally just grind the whole thing, extract the DNA, and then continue. And so we worked hand in hand with curators from the London Natural History Museum to create a minimally morphologically destructive DNA extraction approach, which we called the fly spa. And this whole procedure was published last month. The paper mostly deals with the extraction itself, but what I will be focusing on during this talk is the library prep adaptations that we made to get the most out of the DNA extracts. I will, however, just briefly talk about the extraction method itself. So the key points here are: take an image of your sample at the start, at the end, rehydrate them, because these are extremely brittle, extremely dry. These rehydration process then makes the sample easier to handle, and you can just gently dip it in lysis buffer. Then this lysis buffer undergoes purification and is ready for library prep. But the samples themselves, you prepare using an increasing ethanol concentration wash, as if you were preparing them for electron microscopy. You even do critical point drying, where you exchange the ethanol for liquid CO2 and then just evaporate that. But instead of going for microscopy, you then return them to the collection when they have the 3D morphology completely preserved. Mosquitoes are extremely soft, so you can't just air dry them as you would beetles. And we tested this approach on 4 vector flies. Here you can see the photos before on the left and after on the right for Aedes, Anopheles, Culex, and Glossina, some of the most common disease vectors. And you can see that while there are morphological changes, you are still able to determine by the morphology the species, because all of the species specific characteristics are still there post-extraction. So this was pretty promising. And then we decided, let's just characterize these extracts as if we were characterizing extracts for present day samples, which usually is various methods that measure the amount, the length, the quality of your double-stranded DNA. So on the left here is a picogreen DNA concentration estimate. And hopefully you can see my mouse. If you look at this side of the figure on the right, you have this really wide spread of present-day samples. Please ignore the colors. This is, again, the main point of the paper was the extraction, but just focus on there's dots and triangles and so on. So we have this widespread of DNA amounts that we get from present-day samples, but on average, it's about 200 nanograms per mosquito, because the present-day ones are all mosquitoes. And then if we look at the historic samples plotted here by their collection year, you can see that the vast majority of them are under 50 nanograms total DNA. And these would be considered, at least in our sequencing facility, as borderline unusable samples. So you can give them to the facility, but they will probably fail. Not ideal for us. And then here on the right, we have high sensitivity bioanalyzer chip figures of two of these samples. One is a 1933 samples that has 43 nanograms total DNA. And you can see that most of the DNA is below 200 base pairs. Meanwhile, we have a 1988 sample here that has 206 nanograms. So it's pretty much the same as a present-day sample. And if we look at its DNA length distribution, it's also pretty much the same as a present-day sample. So we have a DNA length peak of about 5,000 base pairs. So of course, this DNA is not running perfectly. It's not being measured perfectly because it implies that, oh, you're giving the machine a perfect double-stranded DNA extract, which this is not. It's full of nicks, it's full of single-stranded overhangs, but at least it gives you an overview of what you're potentially dealing with. And when we saw this, what we saw is, well, this looks like a very bad present-day sample, but that also means that we could technically just use the same approach we do for the present-day samples and modify it a little bit, which our hope was if we keep the library prep as similar as possible, we will also minimize biases downstream once we actually get to population genetic studies. So our thought was, yes, let's just use the same kit that is used for the present-day samples. And the kit that is widely used at our campus is this single tube, double-stranded NebNext Ultra 2 kit, which this is its original protocol. And if you look at the original protocol, it's very clear this will not work for us. So first of all, the adapter ligation step uses bell-shaped adapters with a uracil that should then excise. We don't want that because, again, our DNA is old, and while it might not have a lot of uracils, it definitely has some. And we need to keep them in there. We don't want to basically fragment our old DNA. And the other bit is that during indexing PCR, only one of the primers, in this case P7, has a barcode on it. And we want to keep both sides barcoded, especially since Illumina sequencing technologies are moving more and more towards HiSeq 4000 and NovaSeq. And these technologies have an additional PCR step where it's really important to have barcodes on both sides to make sure that you do not have barcode skipping and potentially cross-contamination in your final pool. But lucky for us, a lot of these optimizations were already done at the Sanger Institute, at the Wellcome Genome Campus, by people from sequencing research and development who have optimized this kit, this double-stranded library prep kit, to work in plates and to be as automated as possible. It's basically all doable on a robot. So you start with sharing your DNA, because this is, again, present day DNA protocol. Adapters are already not the bell-shaped adapters, they're Y-shaped adapters, which is great for us. No need for the additional uracil excision step. And most importantly, both the P7 primer and the P5 primer contain 8 base pair barcodes. The barcodes are quite long. They're usually about 7 base pairs, but the extra 1 base pair on both sides gives you that additional level of security that you will not get primer skipped sequences in your final library files. These libraries are then pulled, quality controlled, and sequenced on your Illumina sequencer of choice. So this has been published by Bronner and Quayle a few years back. And so we just took this and decided, okay, how can we further optimize this for our old samples? Because this works for present day samples that are about 100 to 200 nanograms. So first of all, like I said, we ran all of the samples on the bioanalyzer to see what the lengths of the fragments inside the extracts were. And based on that, we split the extracts into 2 groups. One group was DNA like the one I showed that had 5,000 base pair fragments, so really long DNA. And for those extracts, we decided to split it in half, do 1 sheared and 1 unsheared before library preparation. And for all of the extracts that had very short DNA, we did post-ligation and post-indexing, either a SPRI-B purification or a Minolute purification. And before we actually see the results, another thing that we made sure of is these samples are old. They might not be ancient, but there's still some characteristics that once you get the sequencing data back, you need to take care that you're processing them the correct way. In this case, we couldn't use the present day processing pipeline because it doesn't do adapter trimming. It doesn't do very good even adapter dimer trimming, because for present day samples, you make sure that your DNA is long enough so you don't read through adapters. You make sure that you clean out all of the adapter dimers as much as possible. And for our samples, this is still old DNA. It's very short. We will have all of that. On top of that, most importantly, we want to have some ancient DNA damage patterns to look at and see if we're really looking at old DNA. So we want the fragment lengths, we want C2T and G2A substitutions. And so for all intents and purposes, we used an existing ancient DNA whole genome sequencing pipeline, in our case, eager, which is fully available on GitHub. It's constantly updated. It's relatively user-friendly, and it gives you a really nice summary statistic output at the end, which you can then look at for percent aligning to your reference genome, damage patterns, and other things that you need to say, well, this is a successful library, this is a successful sample or not. And so now we get to the first data plots. And so like I said, the left side is samples that had short DNA. And so we split them in half after library prep. 1 half was purified by SPRI beads. The other half was purified by columns. And on the right-hand side, it's extracts with really long DNA, which was then inputted into the library prep either unsheared or first sheared, purified again, and then inputted. So the circles in these plots represent bead purification and unsheared DNA, so the simpler approach, and then the empty diamonds represent column purified and sheared DNA. And so the first plot is again just picogreen total DNA amount measurements. The really old samples with short DNA are all around 50 nanograms or below, while the really long DNA have also a lot of DNA. So if we look at the percent aligning to the reference genome, we see, first of all, that all of our samples are pretty good. Even samples that here would be considered bad still have 50% aligning to the reference. And these are actually samples that are most distant evolutionary to the reference. So this difference might very well not be microbial contamination, but this is a bad reference for this species. And even if we look at the mean coverage depth, we get about 1x or less for the really low input and short DNA extracts, and much better for the present day extracts. Same with the number of megabases, so how much of the genome is covered, with about 50% in the old samples and closer to all of it in the younger samples. But what is most noticeable here is, like I said, the circles and the diamonds were two different approaches, and there's virtually no difference. So whichever one you choose, you are going to get more or less the same results. And we decided to just continue with the most parsimonious approach going forward, which is we're just going to input unsheared DNA extracts and we're going to spray purify them for simplicity so we don't have to deal with columns. Another thing that we looked at is, of course, like I said, we really want to know about ancient DNA specific characteristics of these extracts because they are old. So again, same colors for the same 10 samples. Dashed lines are the less parsimonious approaches. In the first set of panels, so panel A, you have the fraction of aligned sequences of a specific length. And what you can see here is that with increasing age, seeing as the unsheared versus shear were done on the youngest sample, you also have an increased fraction of 75 base pair reads. So the important thing to note here is this was sequenced on a HiSeq 2500 with 75 paired end chemistry. So you read 75, and then if you have a short insert, those two overlap and you get a single read. The more longer DNA you have, in this case, longer than 139 base pairs, the less overlap you will have and the higher the fraction of the 75 base pair peak will be. And in the case of the really young samples, this is 50%. So we have really long DNA in there. Understandably, samples from, in this case, the 30s and the 40s have much shorter DNA and a much, much smaller 75 base pair peak. And the B and C panels are the 5' C2T substitution and 3' G2A substitution. And when we saw these, we realized the giant mistake we made when we were setting up the initial experiment, which was we completely forgot to check that our polymerase that we used for indexing PCR can actually recognize uracils. And when you look at these figures, you can immediately tell that, no, it definitely can't do that. Because the C2T substitutions, so the side of the molecule that still contains uracil, look awful. But then the 3' G2A, because adenines are added already during library prep, look vaguely better in that you can actually see there is a signal there. So these sequences are deaminated. And so one of in the next set of experiments, one of the most important things we did is that during indexing PCR, we made sure to use the correct polymerase. Luckily for us, Kappa Hi-Fi also makes a Uracil Plus hot start ready mix. So it was easy enough to switch. We used the same one, but without the Uracil Plus for present-day samples. So again, in this round, we decided to, like I said, start with an unsheared DNA extract input, simplifies that step. Make sure we do a bioanalyzer or tape station measurement of the extracts to see what the lengths actually are. And then we can also say if by any chance the library prep failed down the line or not. And what we discovered is that this is a good proxy also for determining the number of PCR cycles that we do. And the thing that we tested in this round actually was the concentrations of spry beads. As some of you are probably aware, the higher the spry bead concentration, the more short molecules you will get. And we had quite a lot of good length molecules, and we don't want to waste too much of the sequencing run of the lane capacity on really short molecules. So we decided to try out using 2.5x spry beads post-ligation and 1.2x spry beads post-indexing versus 2.2x and 2 rounds of 1x, which should get rid of the really short molecules and more adapter dimers. Also at the end, we tried a few different plex numbers for the pools. That just means how many libraries and library blanks you put in the final pool. And so here are the results of that. Again, don't focus on the different colored dots or the different columns. What I want you to focus here are the yellow box and the blue box. So the yellow box was done with the higher spry bead concentration and had a higher plex, so more libraries, more blanks. And the blue box was lower spry concentration, and it was actually 2 pools, but it was about 20 samples and 3 blanks per pool. And so what you can see here is if we just look at the picogreen measurements, again, most of them, besides a few outliers, are around 50 nanograms and below. If we again look at the percent aligning to the reference, in this case, we have 2 Anopheles references, but it's fairly similar to what we saw before. We have samples that are of a more distant species aligning much lower, still about 50%, and then samples that are of the correct species going to 80, 90% aligned. But what is interesting here is if we look at the mean coverage depth and how much megabases of the reference is covered, the samples where we used higher spry beads and a higher plex have very low coverage, have about 1x, and only about 70% of the genome covered, while the ones with lower spry bead concentrations and a lower plex, from just a single lane of sequencing, we are able to get from 2x to 6x coverage, and we cover almost all of the genome. And so in this experiment, we decided to then continue forward for our future mosquito samples to go with selecting against ultra short fragments and to use a lower plex if you plan on sequencing less lanes. If you plan on sequencing many more lanes, you can also go for a higher plex, which is always good for the sequencer anyways, but 20 should be good. And how do we know that this is actually due to the spry bead concentration and not genome sizes or something else? So again, here are ancient DNA characteristics. If we look at the length, the two sample sets in the yellow box were from the 20s and the 40s. And so the one from the 40s, of course, is a bit younger, will have a larger fraction of longer molecules, and so a higher 75 base pair peak. But then the samples in the blue box were both from the 30s. So in between these two, however, our 75 base pair peak is higher in them than in the samples from the 40s. And so we have selected a bit against the really short molecules, increased our fraction of long molecules, which then it then gives us more coverage per lane than before. So a little bit of a selection against the really short molecules is actually really beneficial here. Okay, and if we actually look at the substitution patterns now, they look beautiful. So be sure to use the correct polymerase for your indexing PCR. So if we look at this 5' CTT, it goes up to 4 or 5% at the molecule end. Same with the 3' G2A, and then within the molecule, it stays at about 1 to 2%, which makes sense. That's where you will get some nicks and you will get some deamination before the molecule itself splits. So everything looks fairly good. What we did next is that we had a look at the mitochondrial genome. So we were able to create whole consensus mitochondrial genomes from all samples in both experiments. And this included the samples where we had really bad coverage, nuclear coverage. So it was barely detectable, but even so, as you're all already aware, there's always way more mitochondrial DNA than nuclear DNA. So even our worst sample had a mitochondrial coverage of 10x. Our best mitochondrial coverage was 1,583x. It's slightly excessive. And so we were able to create these consensus mitochondria, and we were able to create maximum likelihood trees, which you can see here for 2 of the Anopheles complexes that we looked at. In red are historic samples. In black are samples that we retrieved from the NCBI database. And so we were able to, based on where these samples group, tell what species or complex they were. Unfortunately, mosquitoes like to hybridize, especially these complexes. And so we are unable for some of them to tell exactly what species they are, but at least we can say, well, it's the correct group. It's the group that is important to us because that's where the major malaria vectors sit. But what is interesting here is you have these 2 samples highlighted in yellow. And these 2 samples had a mismatch in between what their morphology, what they were determined based on their morphology, and what we saw in the mitochondria. And so we were able to reclassify them in their correct group. And this showcases the power of this whole approach to just barcode mitogenomically, so mitochondrially barcode pinned-dried insect collections. And so going back to one of the initial questions, which was insecticide resistance, even though all of these were sequenced on a single lane, we had a look at one of the most common insecticide resistance areas, which is this voltage-gated sodium channel gene. Each row here is a known position where if the mosquito has a variant, it's very likely insecticide resistant, and then each column is one of our samples. So you don't need to focus on it. This is a very busy table, but basically, even at low coverage, all of our samples showed the insecticide susceptible reference variant. So we don't see any insecticide resistance yet. Of course, we would need more coverage, or we would need more samples from these locations and these time points and other time points to make sure that we have the population coverage for that. Or insecticide resistance just started much later than we expected to. Plus, a lot of these samples were actually from the 30s and 40s when there was no insecticide available. But in any case, there is a possibility now for us to go back and look at these samples in the context of our present day populations, also on a much more wider scale of how did the populations change? How do these individuals match their great nieces and nephews and sons and daughters today? And so at the end, I would like to make a list of good practices that we discovered during this project that have been listed also in this talk. So first of all, like I said, this whole protocol, this whole workflow was developed for dried specimens, specifically dried flies. There is a possibility that it will also work on other dry insects, maybe some other dried arthropods, some other dried tissues, I'm not sure. But what I can tell you is they will not work on formalin-fixed samples. We have tried some formalin-fixed tissue, but as some of you working on that type of material might already be aware, formalin causes DNA strands to bind to each other, causes crosslinks, and you really need to boil the sample to release the DNA, which creates single-strand extracts. And you really should probably consider a single-stranded library prep if you have formalin-fixed samples. Another thing that we saw is treat your historic extracts as if they were present-day extracts, like really bad present-day extracts. So do some quality control, even if it's only going to be as if it was all double-stranded DNA. But it's really good to know picogreen measurements, bioanalyzer or tape station measurements to see the DNA that you're dealing with, because based on that, you might want to further adapt the library prep. You might want to keep the really higher spry bead concentration if you have DNA that's mostly under 100 base pairs. And so what I focused on is how we further optimize this single tube, double-stranded Nebnix Ultra 2 kit. We don't do any shearing. We do a 2.2x spray bead post-ligation purification to remove some of the really short molecules. Really be careful that you use the correct polymerase that can recognize urosols for indexing PCR. We do 2 rounds of 1x spray bead purification post PCR, so they can be prepared in plates. It's much easier. It can be automated depending on the number of samples that you have. And this is also good to remove adapter dimers and further select the really short molecules. And if you do plan on sequencing this on a single lane, duplex at the lower number. For our mosquitoes, which have a fairly small genome, about 20 samples is good, but this will very much depend on the size of your genome, because some mosquitoes have much larger genomes. Some insects have also much larger genomes. Plants, I am not even going to get into. They have gigantic genomes. And I would also suggest that you use an ancient DNA pipeline or just an ancient DNA approach to have a look at the ancient DNA damage patterns. Look how your DNA is fragmented, and especially check that you do have some deamination in there. We see deamination even in the samples from the 80s, which look more or less the same as some somewhat old, badly stored present-day samples. And with that, I would like to give a giant thank you to the other members of the Neanderskeeto team. Relevant for this talk, especially, a great thanks to Matthew in red here, who helped. He's from our core bespoke team, and he helped adapt and develop the historic mosquito optimized library preparation protocol. We still don't have an internal fancy name for it. And I hope this was helpful. If you have any questions, please check our paper. There's much more details in there. And feel free to contact me either through my email or my Twitter. And I would like to thank you a lot for listening to this talk.